Okay, so promoter trapping. We're continuing what we went over last time on our topic, talking about how we detect gene function using our reporter genes. So to figure out what genes are responsible for an interesting phenotype, like disease resistance in plants, we can use a method called promoter trapping. And this is only used when we already know what we want our gene to do. So using this method with the GFP and this method with the GUS, that's going to determine like location of where our gene of interest is being expressed, how much of it is being expressed, and when, at what stage of development it's being expressed. We use this promoter trapping method when we already know what we want to figure out. So it basically, we have the reported genes producing their product only in the area of the gene that we're trying to study. So say it's a bit confusing. Say we want to study a plant disease resistance gene. We would expect the disease resistance gene to only be expressed at the site of infection. So if we insert a promoter gene into the plant genome and it's being expressed everywhere, then it's not related to disease resistance because it's not only being expressed at the site of infection. But if we insert a reporter gene into the plant genome and it only gets expressed at the site of infection, well, then we know that that gene probably has something to do with giving the plant disease resistance. So we're then able to study that gene in more detail to figure out more about it. So I'll go over that a bit more slowly now. To figure out what gene is responsible for an interesting phenotype, like disease resistance, we use this promoter trapping technique. In promoter trapping, we randomly insert a reporter gene into an organism's genome without a promoter. So in these last two experiments, we have a gene of interest that we want to know what it does and where it's expressed. We take the promoter from that gene of interest and we put it in front of our reporter gene, and then it gives us this information of where it is, when it's being expressed, what it does. For this one, we're inserting the reporter gene randomly within the plant genome without a promoter. So that means that our reporter gene can only express if it randomly gets inserted downstream of another gene's promoter. When this reporter gene does get randomly inserted downstream of another gene's promoter, if it gets inserted downstream of another gene's promoter, it's going to be transcribed and translated. So we're going to get expression of the reporter gene. If the reporter gene is expressed everywhere in the plant, like we said before, we know it's going to be a constitutive promoter, so it's probably got a function that's the reporter gene's probably been inserted within a gene that has a general function within the plant, so it doesn't have anything to do with disease resistance. We keep doing that. We do this many times. We just basically like randomly throwing darts at the dartboard, hoping that eventually we chuck our reporter gene downstream of the promoter sequence for a disease resistance gene. And then we know that we have succeeded if the reporter gene is only expressed at the site of infection, because this implies that through the random insertion of our reporter gene into the genome, it's landed downstream of a promoter for disease resistance. We then know that the gene downstream from that promoter is involved with disease resistance, and then we're able to study it and learn more about it. This method is a lot faster than the conventional method of isolating a protein, sequencing it, sequencing the DNA, and then comparing genomic structures comparing structure of the gene in a genomic library with different organisms to try and kind of infer what it does. So that was the last topic for the Year 2 genetics module. Hopefully everything makes sense. Yeah, that's it. So I hope you found it useful. Bye.